All right. Thank you, everyone, for joining us for Gina's talk. For most of us, I don't think Gina really needs an introduction, especially not one for me, but I'll do my best. And sort of just on a personal note, I wanted to start by saying thanks, Gina, for doing sort of a lot of work on the DEI stage kind of nationally. I know that the Diversify EEB list was something that you and Meg started here. And that's certainly been something that I think has been super impactful and something I knew about before I even knew that you and Meg were the ones that sort of made this thing. So I think that's really cool. But Gina's work on Morning Glory and its relatives is just super cool and it really expansive. And I'm excited to hear about what you and your group have been working on. So Gina combines morphology and microbial interactions, interactions above the ground, below the ground, competition. And in this talk, we'll hear a little bit about, I think, herbicide resistance, maybe some other kinds of resistance is linked to shifts in mating strategies. So I'll let you take it away.
Okay, thanks, Louise. Appreciate it. Yeah, so thanks for coming to my talk today. I'm going to tell you a bit, kind of an overview of what the lab's been working on, as well as focus in on one particular story that we've been developing on the plant mating system and the evolution of resistance. So like most of you, I'm working at home. Wait, hold on, I'm getting a weird thing from my computer. Unfortunately, because of working at home, you might hear some noise in the background, probably attributed to these two animals, Chris, the anxiety-ridden lab, and then Danny, the very happy-go-lucky Boston Terrier. In case you hear them barking, hopefully you won't, but in case you do, it's always nice to put a face to the sound. So just to give you an idea, kind of broadly what we've been working on in the lab. So basically we focus on ecological genetics and genomics. We think a lot about adaptation to strong selection, and specifically the selective agent that we've chosen to work on is herbicide. It's an extremely strong selective agent designed to kill plants, and it kills about 90% of a weed population. We've been asking in one track of research in the lab questions about the genetic basis and evolution of resistance. So for example, if you have, we take a landscape perspective, and if you have multiple populations, are they resistant due to the same mechanism? Are they resistant due to the same genetic architecture? And even we're trying to drill down and understand if different populations are resistant due to the same mutations. And so this is basically a parallel evolution question. So you have the convergent evolution of the resistant phenotype. Is it due to the same or different changes at the genetic level? I'm not really going to talk about this track so much today, but if you're interested, there's a few papers that we've published over the years thinking about the genetic basis of resistance and associated topics. Another strain of thought in the lab is looking at the mating system and how the mating system could evolve in a regime of strong selection. So many plants will have variability in their mating system. They'll range from outcrossing to selfing, and the common morning glory, which is the plant that we work on the most, it shows it's a mixed mater. So it shows some populations are highly selfing and some populations are highly outcrossing. And so I began to wonder, how does the mating system evolve in a regime of strong selection and adaptation to a selective agent? A relatively new strain of work in the lab right now is thinking about community dynamics under herbicide drift. So generally the herbicide that's been used the most since the early 90s is Roundup, and I'm sure you're all familiar with that. It's used widely because of the use of transgenic Roundup ready crops. And because it's been used at such a high frequency and across many different populations of weeds, it's a very effective weed control agent. Resistance has evolved in a variety of different weed species. And as a result, Monsanto has generated what's known as dicamba ready seeds. And so these, they're crops that are transgenically manipulated so that they are tolerant to both Roundup and the herbicide dicamba. The herbicide dicamba is an auxin-based herbicide, and it's an old herbicide. It's related to Agent Orange, but it went out of use when Roundup started being used more. But it is going to be used a lot now and in the future, really started up about 2017, 2018. It's a problematic herbicide in that it is toxic to humans, and it can drift. So farmers will spray it on their crops, and then it can revolatilize into the air column and move to other areas, unintended areas. So we have a lot of non-target weeds and even horticultural species or plants planted in neighborhoods that are showing signs of dicamba drift. And so this has been on NPR and in the news. You might have heard about it that way. So anyway, we have a couple of grants assessing what are the community dynamics under this herbicide drift. And we're just asking simple questions of, does dicamba drift influence flowering phenology, pollinator presence, and herbivore presence? How is it changing the community dynamics? So other tracks in the lab that we work on, broader themes of plant adaptation have been are generally undertaken by grad students. And a recent graduate from the lab, Sarah Cullum, has worked on character displacement and root traits, which is the idea that you have two closely related species. And of course, we're working on morning glories. If they are found in the same areas and they're competing, their particular traits might evolve to escape that competition. And she's focused on root traits. We have a couple of papers, recent papers from her dissertation. Another track is thinking about leaf shape evolution in a morning glory that you're all aware of, which is the sweet potato, so Ipomea batatas. And Sonal Gupta, as part of her thesis, has been working on leaf shape evolution in this species. And we have a publication in New Phytologist from that, if you're interested. A new track or another track of research in the lab is thinking about trade-offs between resistance to herbicide and herbivory. And two grad students, Nia Johnson and Grace Zhang, are working on this project. And we also have Sasha Bishop, who is working on climate-induced changes in floral traits using temporally collected samples of the common morning glory across the years. And finally, I want to mention Whitney White and Anna Sobel are two other members of the lab, and they're working on the dicamba project. Okay, so that's the broad overview of what we've been working on. Today, I'm going to focus on this right here, thinking about the mating system in a regime of strong selection. Okay, so let me take you back to thinking about agricultural weeds broadly. So agricultural weeds experience strong indirect human-mediated selection. So let's say we have a population here that has variability within this population for resistance to herbicide. We spray an herbicide on the population. I think I said before, herbicides are designed to kill 90% of a weed population, so very strong selection coefficient. And it will reduce the size of the population down through a bottleneck. And individuals that don't survive just due to chance probably survive because they have herbicide resistance alleles. If you continue to use the same herbicide across years on this population, it will rebound, and you will have increased number of individuals in the population over time due to adaptation. And at this point, it's important people study evolution of herbicide resistance in the applied sense, because this represents the loss of efficacy of an herbicide, and it means that a farmer has lost a really important tool for controlling weed infestations. I'm interested in this process because I'm interested in evolution, and it makes a really good study system to study the process of evolution. The things that have been examined in mostly the field of weed science when we're thinking about evolution of herbicide resistance are how many genes control resistance. What's the dominance level of those genes? Are there fitness costs associated with particular herbicide resistance alleles? And another branch of study has looked at gene flow. So what's the possibility that a population has gotten resistance alleles through gene flow from another population? One thing that hasn't been considered is mating system. And the mating system can influence the evolution of resistance, but it's a rarely considered factor. So when I say mating system, what I mean is who mates with whom and how often. So some plants will mate only with themselves, some plants will only mate with others, and some plants will do a variety of the two. The mating system is a really important plant trait because it influences the distribution of genetic variation among populations. It influences plant fitness and the potential to respond to selection, and it influences homozygosity and rates of homozygosity and heterozygosity in progeny, also very important when you're thinking about plant adaptation. So early on in my career, I hypothesized that there would be a relationship between herbicide resistance and the mating system. And basically, if you have a population that's exposed to herbicide application, it goes through population constriction. Individuals that happen to survive due to resistance alleles are going to be in a smaller population. It's going to be constricted. Individuals that have the flexibility so that they can produce progeny from selfing and are herbicide resistant are going to be more likely to be the ones producing progeny for the next generation. So generally, I'm thinking that there's a reproductive assurance argument to be made for a co-association between herbicide resistance and the mating system. And this is really branching off of some of the work that Janis Antoniewicz did when he was looking at the evolution of metal tolerance in the 70s. Another reason that you could expect a relationship between herbicide resistance and the mating system is due to, maybe after resistance has evolved in a population, if you have individuals on sort of the edges of the crops or individuals that haven't gotten or haven't become herbicide resistant because they're not sprayed, they could still be susceptible. And if they are contributing their alleles to an herbicide resistant population, then there would be selective pressure for that resistant population to develop mechanisms to reduce the influx of susceptible alleles. And so there could be selection against outcrossing. Okay, and this is called the prevention of gene flow hypothesis. So with that idea, I have a couple of different specific questions, two different ways that we've looked at this question. Is there a relationship between the mating system and the level of herbicide resistance? And so first we just went out to nature and we looked, right? So we assessed, does the mating system co-vary with resistance in an agricultural weed? So first, what we would want to show is that the level of herbicide resistance across many populations varies across the landscape. As well, the mating system, a plant trait, should vary across the landscape. And then finally, is there a relationship between those two characteristics such that in populations that are more resistant, do they show evidence that they are more selfing? And then we approached it from another way of looking at it, where we took a single population and we were thinking about, okay, once resistance has evolved, how might that then impact mating system evolution? So to assess this, we generated selection lines. We diverged a population to be more resistant and less resistant or more susceptible, and assessed those lineages for the level of inbreeding depression. And I'll talk a little bit more later about why I wanted to look for a relationship between the level of herbicide resistance and inbreeding depression. But just know that for the second part, we took a single population, did divergent artificial evolution, and then did an inbreeding depression study in both the growth chamber and the field. Okay, so the first question, does the mating system of a common weed co-vary with the level of herbicide resistance? And so here's the weed that we work on. This is Ipomea purpurea, the common morning glory. It's a noxious weed found in crops. It employs a mixed mating system, so it's about 50% outcrossing. And you can see here, I'm showing you a flower. It's bumblebee pollinated. And within this flower, the flowers are hermaphrodite, so they have both male and female parts. And you can see right here where my laser pointer is, that's the stigma or the female part. And down here, those are the anthers, that's the male part. The herbicide that we work on is glyphosate, which is the active ingredient in Roundup. Most of you are familiar with this herbicide. It's a non-selective post-emergence herbicide. It accumulates in the apical meristems, and it inhibits plant aromatic amino acid biosynthesis. There's a little bit over 30 weeds that are considered resistant, and this is due to target site and non-target site genetic mechanisms. I'm not going to talk really about the distinction between what target site and non-target site mechanisms are, but we have assessed what the genetic basis of resistance is in the common morning glory. We know that it's a non-target site mechanism, and if you have any questions about that, we just sequenced the morning glory genome, assembled it, annotated it, did a whole genome resequencing, and that's a whole other thing, but you're welcome to ask me about that later if you want. Okay, so first I want to show you just the results of a big study that we did early on. Adam Kuster was a postdoc in the lab, and we assessed if Roundup resistance varied across populations from both the Southeast and the Midwest. So what I'm showing you here is the result of, we went to 54 or so populations across the landscape. We took seeds from maternal lines from each of these populations. We planted them in a very large dose response experiment in the greenhouse. We had about 10,000 plants. And we sprayed them with different levels of herbicide. I'm showing you here the proportion of individuals that survived herbicide at 2 times the field dose. The field dose, if anyone cares, is 1.54 kilograms of active ingredient per hectare. So I'm showing you the results of 2 times that field dose. And what you can see is populations that are labeled in red show high resistance, populations in blue show low resistance. So we have a couple of areas of high resistance in the Southeast, here in Tennessee, and over here in North Carolina. Yeah, so we have high resistance across populations, but still variability for the level of resistance across the landscape. Okay, so the level of resistance varies across populations. Now we wanted to know, does the mating system of this common weed vary among populations? And is there a relationship between the level of herbicide resistance in nature and the mating system? So to assess mating system, we did a progeny array. So I'm showing you here our populations. And the populations labeled in purple, we have estimates of both the mating system and the resistance level. The populations in red, we have information on just the level of resistance. We basically just took these populations that we had screened and randomly chose which populations we would use to look at the mating system. So we took 24 populations, 20 maternal lines within population. We took 10 fruits per line, 1 seed per fruit, and 4,800 individuals to genotype to get an estimate of the mating system across populations. We genotyped 15 microsatellite markers to do this work, and we generated an outcrossing rate, which is notated here by TM, and an inbreeding coefficient. And I'm just showing you on the number of alleles on average were 3.6 per locus. It ranged from 2.8 to 5.5, not super highly polymorphic species, but because we had 15 microsatellite markers, we have a decent exclusion probability and thus decent estimates of mating system across populations. Okay, so what I'm showing you here is the outcome of this mating system work. So I'm showing you the outcrossing rate in the first panel here and the inbreeding coefficient in the second panel. And over down here, we just have population number. And so what you can see is that populations varied for their level of outcrossing. It ranged from 0.2 to 0.8. On average, the level of outcrossing in this species is 0.5. And so effectively, that's a pretty big range. So species that have an outcrossing rate of 0.2 are considered highly selfing, and species that have an outcrossing rate of 0.8 are considered highly outcrossing. So I think I said 0.2 highly selfing, 0.8 highly outcrossing, right? Okay, so on average, across all populations, we have evidence that this plant is a mixed mater, but there's a lot of variability across populations. The inbreeding coefficients of the moms, 12 of 24 populations showed significant inbreeding coefficients across populations. Okay, and so the main question, is there a relationship between the level of herbicide resistance and the outcrossing rate? And what I'm showing you here on the the x-axis is the level of resistance across populations. As you're over here, you're highly resistant. Over here, you're low resistant. And then on the y-axis, I'm showing you the outcrossing rate. And we can see that there's a strong negative relationship between outcrossing rate and the level of resistance. So the more resistant populations were selfing more often, okay, just as predicted. And the maternal inbreeding coefficients were positively related to resistance, as you would expect. Okay, so we have a negative relationship between the outcrossing rate and the level of resistance, suggesting that highly resistant populations tend to self more often. So I began to wonder, is high resistance, high selfing a stable strategy? Okay, so what's going on with these high resistant, high selfing populations? Well, then we have to think about what are the factors that could influence this relationship between the two. So one, we could have, the relationship could be due to environmental or genetic covariance, right? I don't think that it's an environmental relationship that I'm showing you here. Basically, although we did collect all the populations from nature, We have so many populations that it would be strange if it weren't genetic covariation. But to be honest, I can't rule that out. The other thing that could influence the relationship between high resistance and high selfing is inbreeding depression. So if those high resistant populations that self more often, if those individuals show inbreeding depression, then over time you would expect that the mating system should evolve to be more outcrossing. So eventually it would become more resistant and more outcrossing. So then from that line of thought, I wanted to assess what's the level of inbreeding depression across lineages that are showing variable levels of herbicide resistance. Okay, so is inbreeding depression a factor, right? Is there any evidence for inbreeding depression in highly resistant individuals or populations? And so this is a decent question to ask specifically with this species, because one of the initial species that showed that what inbreeding depression even was the common morning glory. So Darwin took the common morning glory, and because it's such a labile genetic model organism, he was using it as a model way back when. He basically selfed and produced seeds from selfing and outcrossing for 10 generations. And you can see here at the end of the 10th generation, individuals that were produced by selfing were shorter than individuals that were produced by outcrossing. So there's inbreeding depression in this species, and it was shown back by Darwin. Okay. So now we're on to this part 2, how might resistance impact mating system evolution? So if we have the evolution of resistance, is inbreeding depression going to influence the mating system from there on? And so to do this, we generated selection lines and put them into a growth chamber study, and as well assessed if there is evidence of inbreeding depression in the field. Okay, so let me tell you a little bit about my design to artificial selection to create resistance lines. So we took a single population that was collected back in 2003, and we sprayed herbicide on this population. We took pre-spray height and post-spray height measurements. And so we effectively took a height before divided by the height after to figure out which individuals were more or least resistant. We took individuals that showed 20% of the initial population that showed the least dieback after being sprayed, and 20% of individuals that showed the most dieback after being sprayed, okay, to make our most resistant and least resistant lineages. And we took a random control. So we took individuals just randomly from this population as a control. We took these individuals and we did artificial selection. So I'm showing you here, we had 2 different diallels, which just means two sets of crosses that are independent per direction of selection. So we had 2 diallels of selection going up for increased resistance, 2 diallels of decreased resistance, and 2 diallels that were just controls. Okay, so we did all these crosses, we generated progeny, we sprayed the progeny again, and then we measured height before and height after being sprayed. We did this for 3 generations to develop our resistant lineages, our increased susceptibility lineages, as well as our random controls. And so you do these random controls because you want to control for drift, right? So we've got a couple different ways of controlling for drift in this artificial selection experiment. One is the use of 2 different diallels per direction of selection, and the other is using a random control. That's kind of more information than you need. Okay, so then at the end of these 3 generations of artificial selection, we then generated progeny produced by both inbreeding and outcrossing, because ultimately we wanted to assess inbreeding depression, right? So we took, I'm showing you here my maternal lines. I'm labeling them R1 through R6. So pretend like we have 6 resistant maternal lines that are separate here. We selfed those maternal lines. Then we took each of those maternal lines and we crossed them to other resistant individuals to make outcross progeny. Okay, so at the end of this, so we did 3 generations of artificial selection to diverge the level of resistance in the population, and then we both outcrossed the progeny and selfed them. So we have 6 lines of each selection type. So we've got resistant, susceptibles, and controls, and we've got 2 different breeding types. We've got produced by selfing and outcrossing. Okay. So then we did a growth chamber experiment with these artificially diverged lineages to look to, first of all, did we diverge the level of resistance in this single population, right? We started with a single population at the beginning. So we put 18 families, so it'd be 6 families each that were increased resistant, decreased resistance, which are the susceptibles and the controls. We took both self progeny from these lines and outcross progenies of these lines, put them into the presence of the field dose of herbicide, twice that, and then an environment with no herbicide. We phenotyped them after applying herbicide. We took a qualitative score, which was alive, almost dead, and dead. And then we also dried them at the end of the experiment to get biomass. Okay, so now I'm going to show you the results of this artificial selection scheme. Were there any differences between selection lines? Yes, there were. So I'm showing you on the y-axis. You can't see when I'm pointing with my hands. Let me remember to use the pointer. So I'm showing you the percent alive on the y-axis, and I'm showing you the average, excuse me, for the control, non-selected lineages, the resistant lineages, and the increased susceptibility lineages. And you can see that at 1.5 and 3 kilograms, so 1 times the dose of the field rate, and then twice that, we have a response where we have the resistant individuals had more individuals in that were living compared to the controls and the susceptibles at both doses of herbicide. Okay, so in terms of death, we had a response from the artificial selection. We've moved the population so that the increased resistant lines are showing more resistance. The decreased resistant lines are showing more susceptibility. Okay, and in terms of dried biomass, you see the same general trend. I'm showing you the controls in off-white, the resistant, increased resistant in red, and the increased susceptibilities in blue. And what you can see at the 1.5 or the field dose of herbicide and at the higher dose of herbicide, in terms of dried biomass, we tend to have a higher dried biomass of the resistant individuals compared to the susceptible individuals and sometimes the controls. Okay, so basically this is just to show you that our 3 generations of artificial selection diverged the population. Okay, so now I told you that we put progeny in this experiment that had been produced by both inbreeding or by selfing and by outcrossing. So let's look at the data according to breeding type. Okay, so I'm showing you here dried biomass on the y-axis, and I'm showing you the average for the control or the non-selected individuals, the increased resistant individuals, and the increased susceptibility individuals, okay? And the white is now showing you the average for individuals produced by inbreeding, and the red is showing you the average for individuals produced by outcrossing. And what you can see is that there's a breeding type effect. Okay, so generally in the control and the susceptible lineages, the individuals that were outcrossed showed higher dried biomass than individuals that were produced by inbreeding. But you don't see this effect for the resistant individuals, and that's going to be important later. Okay, so and at the higher dose of herbicide, what we see is a significant selection line by breeding interaction. So we still see that the outcrossed individuals from the control and susceptible lines do better in terms of dried biomass, whereas the resistant individuals show the opposite relationship, where the inbred individuals are showing higher dried biomass compared to individuals in the resistant lineages produced by outcrossing. Okay, so that explains that significant selection line by breeding interaction. So the inbred individuals of the resistant lines at the higher dose of herbicide are really doing better than the resistant individuals produced by outcrossing. Okay, and so that's what we see in the controls and the susceptibles are evidence for inbreeding depression. What we see in the resistant lineages is evidence for outbreeding depression. Okay, so this was just a growth chamber study. We can't really assess fitness very well in the growth chamber. So we wanted to assess, is there evidence of inbreeding depression in the field? So we took our experimental design and put it out in the field. So I'm showing you here, we took 18 families 6 each that were increased resistance, increased susceptibility, and control lineages. We took their selfed and outcrossed progeny. We planted them into the field in an array like this, and we let them grow for a bit, and we applied a low dose of herbicide, which is about half the field dose. We just wanted to stress them out in this experiment, but we didn't really want to kill anyone. We wanted to assess fitness. And then we also had them in an environment of no herbicide. And so in total, we planted a little bit over 1,400 individuals. We took a bunch of phenotyping on these plants. So we took height before herbicide, height 3 weeks after herbicide, the day of first flower, total number of flowers, total seed produced. And of those seeds that were produced in the field, we took those back to the lab, and then we did germination assays. How well did the seeds that were produced in this experiment also germinate? Okay, and so we have a lot of data, but what I'm going to focus on is going to be the total seed produced as well as their germination. So I'm showing you here in the absence of herbicide, we're going to look specifically for the presence of inbreeding depression, okay? And so I've got total seed by individuals produced in the absence of herbicide on the y-axis. And in white, you've got the data for inbred individuals. In red, you've got data for the outcrossed individuals. And you can see that there's a significant breeding and type effect. Effectively, the outcrossed individuals produced by outcrossing produced more seed than individuals produced by inbreeding. So across these lineages in the field, there's evidence for inbreeding depression. In the presence of herbicide, we still have a breeding type effect. We can see that individuals produced by outcrossing in the control and the susceptible lineages produced more total number of seed than the inbred individuals, but you don't really see this effect for the resistant lineages. Okay. So there's a way that we can look at inbreeding depression by summarizing it by a metric called delta. And so I'm showing you here, delta is on the y-axis, and this is 1 minus the number of seeds from inbred individuals divided by number of seeds from outcrossed individuals. And over here, just I'm showing you the y-axis, if you're more up here in this part of the graph, that means that outcrossed individuals are doing better. If you're down here on the graph, on the y-axis, that means inbred individuals are doing better. And so this is just a quick way to summarize what I've shown you in the previous slides. And I'm showing you here for, let's see, the control individuals, the resistant individuals, the susceptible individuals. In the absence of herbicide and the presence of herbicide, we have evidence inbreeding depression for the controls, the non-selected controls, as well as the increased susceptibility lineages. But we have no evidence for inbreeding depression in our increased resistant lineages for the total number of seed. Okay, so now let's look at germination. So I told you that we took those seeds from the field, brought them back to the lab, and then looked at the proportion of those seeds per plant that germinated. And so this is really just a slide for me to remind myself to tell you that in the absence of herbicide, we typically have around 60% germination of those seeds from the field. This looks like it's significantly different. There's a selection line effect, but there's not. And in the presence of herbicide of the seeds that you collect from the field, we have about 30% germination. So there's an effect generally of the herbicide on seed germination of individuals that are producing seed in the field. Let's look at this in terms of inbreeding depression. So I'm showing you here the proportion of seed germination on the y-axis. We're summarizing it again for the controls, the resistant lineages, and the susceptible lineages. And what you can see is that there's a significant breeding type effect, meaning that individuals in the absence of herbicide that were produced by outcrossing have significantly more seeds that are able to germinate than individuals that were produced by inbreeding. Okay, so there's inbreeding depression in the proportion of seeds that will germinate that were produced in the field. Now, this is in the absence of herbicide. In the presence of herbicide, there's still a breeding type effect. So there's still evidence that there's something going on, but different between the inbred and outcrossed individuals. But there's a significant breeding type by selection line effect, okay? And what I want to point you to is that resistant individuals, when sprayed with herbicide, inbred individuals were produced seed that germinated more often than outcrossed individuals, okay? So there's evidence for outbreeding depression in the presence of herbicide for those individuals that are from resistant lineages. And you don't see this in the control or susceptible lineages, okay? So another way to look at this is by summarizing it with delta. And again, over here on the y-axis, I'm showing you delta. If you're up here, outcrossed individuals do better. If you're down here in this part of the graph, then inbred individuals do better. And what we can see quite easily is that for our resistant selection lines, individuals that were inbred did better, produced seeds that germinated more often than individuals that were produced by outcrossing. So evidence for outbreeding depression. Okay, so to summarize this part of the talk, resistant inbreds seem to do better in herbicide, okay, than outcrossed individuals. So in our growth chamber study, so we're showing this not only in the field, but we're showing it across different experiments and different metrics of plant performance. Okay, so in the growth chamber, in the lower dose of herbicide for dried biomass, in the resistant lineages, we showed no evidence for inbreeding depression, where the controls and susceptibles did. In the really strong selection, right, so 2 times the field dose, we're showing evidence of outbreeding depression for dried biomass in the resistant lineages, where the controls and susceptibles are showing inbreeding depression. In the field study, we showed no evidence of inbreeding depression in the resistant lineages, although we do see that in the control and susceptible lineages. And in terms of proportion seed germination, we find evidence for outbreeding depression in the resistant lineages, where the inbreds are doing better in terms of germination compared to the resistant outcrossed individuals. So that's kind of an interesting, weird result. So what are the genetic underpinnings of this? So I have a few ideas of what could be going on. So perhaps there's underdominance in loci that are tightly linked to resistance. And just to remind you, to make our outcross progeny for these experiments, we took our resistant lineages, or all the lineages, and outcrossed them to 3 different individuals. So it could be that perhaps loci that travel or are strongly linked to resistant loci show effectively higher fitness in the homozygous state compared to the heterozygous state, so that would be underdominance. There could also be Dobzhansky-Mueller-like interactions or epistasis. So perhaps a resistance locus or loci interact with alleles of loci from other genetic backgrounds, decreasing fitness. Okay, so just a couple of ideas of what we're thinking could be going on. And finally, one other part of the study that I want to show you. So we're talking mostly in what we've done up to this point is mostly phenotypic in nature. So what's going on with the evolution of phenotypes. We did do a transcriptome experiment to understand the transcriptomic basis of inbreeding depression, as well as the interaction between the level of resistance and inbreeding. And so we took our families that were in the field. So for this experiment, we took 12 families, 4 each that were from the resistant, susceptible, or control lineages. We took leaf tissue from both self and outcrossed individuals that were in the presence of herbicide and the absence of herbicide. And we assessed gene expression in these individuals at both 8 hours post herbicide application and 36 hours post herbicide application. And that should actually say 32 hours. So here's our experimental design. We had 4 families, 3 selection lines, 2 crossing treatments, 2 spray treatments, 2 time points. So effectively, we sequenced 96 transcriptome libraries. We generated 7 lanes of Illumina sequencing. This was about 88 gigabases from sequencing, and we had about 28 million reads per individual, which was probably overkill. Okay. So we wanted to assess, is there differentially expressed genes associated with inbreeding? To do this, we used, I'm just going to show you the analysis that we did using weighted correlation network analysis, or WGCNA. It describes correlation patterns among genes from an expression array, whether it's gene expression from RNA-seq or from microarrays. It finds clusters of highly correlated genes, and then you can relate these clusters to a particular trait. And so what I'm going to focus on today, although we have a lot of different effects that I could talk about, I'm just going to talk about clusters of gene expression associated with selection line and breeding types from our experiment. The specific comparisons that I'm going to talk about are only in the herbicide environment, and we're going to look at control inbred individuals versus control outcrossed individuals. And just to remind you, control means non-selected, okay? Then we're going to compare resistant inbred versus resistant outcrossed individuals, and susceptible inbred versus susceptible outcrossed individuals. Okay, so to ask this, so first I want to show you a graph, and I want to explain this graph to you. So I'm showing on the y-axis mean eigengene expression, which effectively is the expression of correlated clusters of differentially expressed genes. Okay, so it's a cluster of genes. The off-white dots that I'm showing you here are inbred expression levels. The red dots are the outcrossed individuals expression level. And then I've summarized it according to control, resistant, and susceptible selection line. And over here, I'm showing it according to 3 different clusters in this example. And the cluster names are modules of correlated genes. So we've got saddle brown, sienna 3, and tan. We had a reviewer say recently, change those names. I really don't like them. And we said, we agree, we don't like them either, but that's what the program does. So we're going to use them. Anyway, so what I want to show you is effectively at 32 hours post herbicide, we have differences in gene expression of the outcrossed and inbred individuals, but only really from the resistant lines. Okay, so here in terms of saddle brown gene expression cluster, you can see that the outcrossed individuals had higher expression than inbred individuals. And so I'm putting it in blue for meaning effectively what's going on with the inbred individuals, right? So inbred individuals showed lower expression of nicotinamide metabolism, divalent metal ion transport, and gametophyte development. In this sienna 3, this is the cluster that I'm most interested in because it corresponds similarly to our phenotypic results, right? So we see in this sienna 3, higher gene expression of inbred individuals relative to outcrossed individuals in our resistant lineages. And what's in that cluster is DNA strand elongation involved in DNA replication, steroid homeostasis, and cell cycle process. So effectively what this is showing is that at 32 hours post herbicide, those inbred resistant individuals are involved in cell cycle process. They're just sort of humming along and doing, involved in DNA replication compared to the outcrossed individuals, which I think is interesting. And then in the tan cluster, we see that the resistant individuals, the outcrossed individuals are showing higher gene expression for response to UVB compared to the inbred individuals. Okay, so to summarize across these 2 experiments, first we were asking, does the mating system covary with resistance in an agricultural weed? And we found that yes, it does. As the populations across the landscape show higher levels of herbicide resistance, they tend to self more often. And then we wanted to sort of turn a little bit and ask the question in a different way. How might resistance impact the mating system? And is there evidence for inbreeding depression associated with herbicide resistance? Interestingly, across different lineages created by artificial selection, we find that the resistant lineages show no evidence for inbreeding depression. And in fact, in a couple different metrics show evidence for outbreeding depression. So those resistant inbreds are doing better. And what this would mean if the single population dynamics looking at inbreeding depression are repeated across the landscape, this would mean because our resistant inbreds did better than our resistant outcrossed individuals, then a selfing mating system and resistance should remain associated over time. Okay. So what does this mean more broadly? What does associations between the level of herbicide resistance and the mating system mean for biocontrol in the context of mating system evolution and for plant adaptation more broadly? So for applied biocontrol, you could think that if a population of weeds evolves to become highly resistant and also evolves a highly selfing mating system, if a farmer or weed scientist or whatever wanted to take a strategy of planting susceptible individuals in the margins around this resistant population to kind of act as a reservoir of susceptible alleles, if the plant is already showing a high selfing mating system or tends to self more often, that strategy is not going to be effective, right? In practice, no one uses reservoirs of susceptible populations of weeds to control resistance. Farmers just spray different herbicides. In the context of mating system evolution, we're always interested in what components of the environment are influencing mating system evolution in plants. We see a lot of variability across species and even within species. And so this is one example where you have an environmental correlate or adaptation showing a reason for differences of mating system across populations. And what would this mean for plant adaptation more broadly? So you can think, well, plant populations are being exposed to lots of different strong selective agents. And if this is a model system for other types of strong selective agents, such as temperature extremes or et cetera, having to do with climate change, then if plants are going to evolve higher levels of mating for perhaps reproductive assurance, then if they needed other alleles that they would get from outcrossing just by chance to be able to respond to a new selective agent, they would not be able to get those important reservoirs of genetic variation. Okay, so with that, that's the end of my talk. I would like to acknowledge all of the postdocs and grad students that have worked on this topic. So Adam Kuster, I'm showing him right here. He did pretty much all of the mating system work at the beginning of the talk. Megan Van Atten came in later with Anna Sobel and worked on the results of the artificial selection and looking for inbreeding depression and the RNA-seq experiment in the field. And other grad students and postdocs, as well as funding from USDA, NSF, and University of Michigan. And I think that's it. So I would be happy to take any questions that you might have.
That's great. Thank you. And if there are any questions, feel free to pop them in the chat or raise hands in Zoom, and I'll try and keep on top of that. Are people able to unmute themselves, or is that something we'll have to do? I don't know. We'll figure it out. Maybe while we're waiting for some people to compose their questions, I'll ask one. So is there any evidence, or do you have a feeling? I think you might have shown one slide that showed this, but other herbicides have similar effects on mating systems?
No, so this is the first example looking at plant adaptation and the plant mating system.
Yeah, I'm wondering, so if you find herbicides that have the opposite effect, can you combine these things and create like a cocktail herbicide that kind of uses evolution against itself to control resistance?
I mean, yeah, I would think like you would, if it would work that way, and I don't know that it would work that way, but if it would work that way, then you'd want to rotate them, right? So you'd be selecting for different strategies of resistance. Well, I think if you're going to use a different herbicide, you're just going to like basically get rid of that population. And so, yeah, I have no idea.
Yeah, there's some interesting work with antibiotic resistance and microbes that be fun to chat about sometime. Still waiting. Don't see any hands up. Got more questions.
I'll throw one out there. This is Tim, your old pal. Nice to see you. So when you did this experiment, you did like the selection lines. You did like 3 generations, I think. You see change over that period, or yeah, I'm just wondering like why you chose 3 or what expected it to kind of increase over time.
Yeah, so we chose 3 basically because it was taking longer than expected to do the artificial selection, and we needed to get some results from the field, right? And so I wanted to initially do 8 generations of selection, but we did see a response after 2, and I decided to do 1 more generation. And so I did that 3rd generation, and it's kind of interesting. In the growth chamber, we saw difference across the lineages. This was before we ever put them out into the field. But then we also saw that there was only differences between the selection line. There was no longer any variation within the families within selection line. So I figured at that point, at time point 3, we were done with genetic variation, right? And so that's another reason why we just decided to go ahead. Okay, we've taken this one population and we've done the extent to which the response is going to happen in this single population. And so we just decided, well, we've caused some movement. Let's put them out in the field and then do some work. And then in subsequent field experiments, we still find genetic variation across the maternal lines for response traits, right? So I probably could continue the selection if I wanted to. Yeah, so it's a bit of practicality, and then thinking I hit a wall, and then realizing, in different environments, you're going to see the expression of different, genetic variation. So, yeah.
We go to John.
Yeah, hi, Gina. Great talk. It was really interesting. It was nice to see what your whole lab is doing, and it was a really wonderful study. I have sort of a predictive question. What do you think is going to happen then with the new dicamba stuff? You see some of this going to repeat itself or anything? What?
Yeah, I mean, that's kind of similar to Luis's question. And when I was originally thinking about the use of dicamba, I was thinking, well, my study system's gone because the morning glories are just all kind of, go extinct. And so, we'll see in a few years, like, are the populations that we've been studying since 2003 still going to be there following dicamba use? And I guess, there's 2 ways I can answer this. Like, one, if it really is a reproductive assurance argument, right? So individuals that survive strong selection and then can produce seed are going to be more likely to also have those selfing alleles, then it should work, right? Regardless of herbicide, assuming the strength of selection is the same, right? And that's another thing, this is a tangent. That's one thing that I haven't really played with too much, is how do these plant populations and the mating system respond when I vary that selection coefficient, right? By varying the amount of spray. The other thing, though, that could come into play is maybe it's not a reproductive assurance argument. Maybe loci that are related to mating system and resistance just happen to be strongly linked together, or in close physical proximity, right? And in that case, if you're selecting on different components of genetic variation, it's just going to be due to chance what other alleles are close by and are going to be hijacked with the resistance. Yeah, nice question.
Catherine has a question.
Yeah, thank you. Thanks, Gina, for a nice talk. And my question is maybe a little bit like John's, except in a slightly different direction. Are there biological controls that are not herbicide for controlling the weeds in this genus? And if so, would those control agents also affect mating system, or do you have any evidence for that?
Yeah, so I don't know any control agents. The one things that they don't do super well with is if they're in competition with grasses. So, I mean, if you wanted to come in and plant generation or the next sort of crop rotation with really tightly, densely packed grass, I think the morning glories would not do so well. The problem with morning glories specifically is that they can seed bank for up to 20 years. So if you have a population, it's just going to be in the seed bank for quite a while. And so I don't really know if any. We have a couple of rust funguses down south that impact the morning glories. Whether or not they would be an effective biocontrol, I don't know for sure. They get some herbivory, but not a ton.
Okay, thanks.
Yeah, sure.
Hey, Gina, or YQ.
Huh?
I have a general question. What's the worst consequence to the yield of the crops if we don't do anything with this morning glory weed?
Yeah, Yeah, that's a great question. So I mean, I've just read, I haven't done any of this sort of work, but I've read that morning glory infestations can reduce yield of soy crops by 80%. So yeah, you can have a pretty significant loss of your crop yield if you don't control them.
Okay, thank you.
Yeah.
John has another question.
Yeah, I just want to comment that they can reduce the yield of coffee in Puerto Rico by 100% very easily. And I just want to tell you, Gina, I hate your plant.
So many people do. You are not alone. The greenhouse people at Georgia just really, I mean, they liked me personally, but they did not like the morning glories. I often brought them tacos to make up for it.
So, I have another sort of a broader general question. Getting all these talk about the climate change, so we're all switching to, say, carbon zero economy. But, for all these pesticides, herbicides, and, what is our prospect of reducing use of those chemicals in agriculture?
Yeah, so yeah, their use in production does matter quite significantly for the carbon footprint. I don't know. That's a really big question, Mike. I don't know that I can answer it. It's a good question, but yeah. So you got me.
Great. If there are no more questions, maybe one more round of applause for Gina. Thanks again for a great seminar.
Yay. Thanks, everyone. Bye.